cytotoxicity activities and growth promoters of animals and plants. So it has vast application and different purpose. It can be used as pharmacological products, or can be used as industrial usages, or can be used for waste recycling. And then we have so many important applications towards the microbial enzymes. The next one we will move on to the marine organisms. Oceans and marine organisms. So the marine ecosystem covers about 70% of the Earth's surface, and marine environment provides an extremely complex with wide range of microbes.
Subham, how can we take the attendance from here? Subham, how can we take the attendance from here?
Zoom? Manually.
Manually you have to take.
Manually at when you close the meeting, there could be a
Yes, that I know. That we have to copy that and then we got to manually we have to. So good morning. Am I audible? Good morning. Am I audible to you? Okay. So, good morning, everyone. And this is the first class in this semester. I think, hope you all know me very well. I think you don't want to introduce myself. Anybody wants my introduction? Anybody wants my introduction? Hope you know. Yes. You know me very well. Right. So this semester I'm going to handle virology. Yes, we know you. Yes, Aninder Kaur. I think last semester I have sent a lot of prizes. So this year also, this semester also, those who are very active, you can get immediate instant prizes in your account. The same thing will be followed. So today we are going to discuss the orientation to virology and what is virus. Still you are joining. Virology is nothing but the study of viruses. So the study of viruses is said to be virology. So any word which ends with logy, L-O-G-Y, if any word end with logy means study. Logy means study. It's a Greek word actually. Yes. The study of viruses. So what is a virus? What is a virus? What is the property? This thing we are going to see now. Can you see the screen? Yes. So coming to viruses. What are viruses? The viruses are host-specific, obligate, intracellular parasite, which are ultra microscopic in nature, measures in nanometer. The viruses are host-specific, intracellular parasite, which are ultra microscopic in nature, measures in nanometers. So what do you mean by host specific? So viruses are, you know that they are intracellular parasite. That means they can grow only inside a cell. So if you take in case of bacteria, bacteria can grow in anywhere. If a bacteria present in a water, it can grow easily. So it can utilize the nutrient present in water and it can grow. If a bacteria present in soil, it can take the nutrients from soil and it can grow. Whereas viruses cannot grow when they are outside of a cell. So when they are present inside a cell, the cell may be a plant cell or animal cell or a human cell or a bacterial cell. So it requires a cell, host cell. So that's why we mentioning as host specific obligate intracellular parasite. That means it strictly adapt to the parasite. Sorry, it strictly adapt to the host. So without host, the virus cannot grow. So if the virus has to grow, it requires a cell, a living cell. It requires a living cell. That cell may be a plant cell or animal cell or a human cell, or it may be a bacterial cell. So that's why viruses are specific obligate intracellular parasite. Intra means inside. Inside the cell. Then obligate means strictly. Obligate means strictly. So it should be present in the cell, then only it can grow. Otherwise, outside of the cell, it cannot grow. Then these are all ultra microscopic in nature. What is ultra microscopic? See, if you take bacteria, it is microscopic. Microscopic means it can be measured in a micrometer. See, the size of the bacteria is 5 micrometer, less than 5 micrometers. Just take 1 micron. So 1 micron, the size of the bacteria will be 1 micrometer, and that can be visible under normal laboratory microscope. But in case of viruses, they are ultra microscopic. That means it is 1000 times smaller than a bacteria. 1000 times smaller than a bacteria. So it cannot be easily visible under a normal microscope where bacteria can be seen. So that's why they are mentioned as ultra microscopic in nature. 1000 times smaller than bacteria. That means they are measured in nanometers. So they are measured in nanometers. 20 nanometer, 200 nanometer. What is the size of nanometer? It is 1000 times smaller than a micrometer. 1000 times smaller than a micrometer. That is a nanometer. Okay. So the viruses are strictly adapt to the host, and it can live only inside the host cell. So they are obligate intracellular parasite, and they are ultra microscopic in nature, and it is measured in nanometers. So coming to the properties of viruses. So coming to the general properties of viruses, these are smaller than bacteria. They are smaller than bacteria. As I said, the size ranges is 20 to 200 nanometers. So they are 1000 times smaller than a bacteria. The viruses are 1000 times smaller than a bacteria. The size range will be 20 to 200 nanometer. They are acellular. They are acellular. That means they does not have a cell wall. Like bacteria, bacteria has a definite cell wall. The cell wall is composed of polysaccharide or peptoglycan. But in case of viruses, in case of viruses, viruses has no cell wall. So they are acellular. But it depends on host cell for its multiplication. So it cannot multiply individually. It cannot multiply individually. If you take bacteria, I will compare bacteria and virus. Bacteria can grow and multiply independently. That means if a bacteria present in water, it can easily multiply in the water by taking the nutrients present in the water. If bacteria present in soil, it can easily multiply by taking the nutrients present in the soil. If bacteria present in any other place, so it can easily multiply without depending on any cell. But in case of viruses, for their multiplication, it solely depends on host cell. The host cell may be a plant cell. It may be a plant cell, it may be animal cell, or it may be a human cell. But it requires a host cell for its multiplication. So when it is present inside a host cell, they can multiply. Otherwise, they cannot multiply. So they are acellular. They depends on host cell for its multiplication. Then they are host specific. So they are host specific, able to multiply only inside the specific host. So similar to when compared to bacteria are not host specific, but viruses are host specific. What do you mean by host specific in the sense? See, if you take bacteria, if it is present in water, just for bacteria, if it is present in water, it can multiply there. Bacteria is present, when present in water, it can multiply there. When the bacteria enters into a human host, the bacteria can also multiply inside the human host also. Once again, once the bacteria from human cell, it goes to water, there it can also multiply. Once a bacteria present in soil or in sewage, it can multiply there. When it reaches a host cell, then it can multiply inside the host. Then once it excreted from human host, when once again it goes to the sewage, then it can multiply. So they are not host specific. They can multiply everywhere. But viruses are host specific. That means human virus can multiply only human beings. So those viruses which multiply inside the human cell can multiply only inside the human being. It cannot infect birds. It cannot infect animals. It cannot infect plants. Similarly, the plant virus cannot infect human beings. So those viruses which cause diseases or infect in plant cell, the viruses which cause infection in plant cell cannot multiply inside the human cell. And those viruses which multiply inside the human cell cannot multiply in bird cell. And those viruses which multiply inside the bird's cell cannot multiply inside the bacterial cell. So they are host specific. If it is a human virus, it is only multiply inside the human virus. If it is a bacterial virus, it can only multiply inside the bacterial virus. If it is animal virus, it can multiply only inside the animal cell. If it is a plant virus, it can multiply only inside the plant cell. So they are host specific. It depends on the host. So it can multiply only inside the specific host. Yes, Anidhar Kaur, yes, correct. Coronavirus only affect the human, not animal. Yes. Coronavirus cannot infect plants. Coronavirus cannot infect bacteria. So they are host specific. Done. Just you take, they contain either DNA or RNA as its genome, but not both. See, the viruses may contain either any one. DNA may present or RNA may be present. But DNA or RNA cannot be combined, cannot be present in a single viral cell. But if you take bacteria, DNA and RNA both present. If you take bacteria has both DNA and RNA. But in case of virus, any one, either DNA will be present or RNA will be present. So those viruses which has a DNA, we classify them into DNA viruses. and those virus which has RNA as the genetic material, we classify them under RNA viruses. So based on the nucleic acid composition, we can classify the viruses into DNA viruses or RNA viruses. But the DNA and RNA cannot be combined and present in a single viral cell. That they may be enveloped or non-enveloped. They may be enveloped or non-enveloped. That means it is like similar to a cell wall, outer covering. Envelope is the outer covering. Sometimes some viruses may has envelope, that is outer covering. And sometimes it may be non-envelope. Some viruses does not have any envelope. But whatever, if it is the enveloped viruses, the envelope is derived from the host cell. It does not have the envelope on its own. It does not have anything on its own. So whatever the virus has, it is derived from the host cell. Now, they can be observed only through electron microscope. They can be observed only through electron microscope. As I already said, they are measured in nanometers. The viruses are measured only through, they are measured in nanometers. Their size is ultra small. Their size is too small. That is, it is 1,000 times smaller than a bacteria. So they cannot be seen with normal laboratory bright field microscope. They cannot be seen with the normal laboratory microscope. So they can be seen or observed only through the powerful electron microscope. So normally they cannot be visible under normal laboratory microscope. They can be visible only through electron microscope. They cannot be cultivated in ordinary chemical media. They cannot be cultivated in ordinary chemical media. That means if you take bacteria, bacteria can be cultivated in bacteriological culture media, where we are using lot of media we are using. See, nutrient agar, mechanical agar, eosin methylene blue agar, and TCBS agar, mannitol salt agar, and then cysteine agar, then cled agar, then cetrimide agar, herilla agar. So a lot of medias we are using for the cultivation of bacteria, even blood agar like that. So they are chemical media actually. All the composition, if you take nutrient agar, it contains peptone, sodium chloride, then glucose, yeast extract, beef extract, like that. If you take eosin, it contains eosin, beef extract, like that. Salmonella shigella agar, whatever the media you are using, they are chemically synthesized. So the viruses cannot be grown. The virus cannot grow in these type of chemical medias. The virus cannot grow in these type of chemical media. So they cannot be cultivated in ordinary chemical media. So they can be cultivated only in living media. That means they are, as the viruses are obligate intracellular parasite, they can only live inside the cell. So we need cell culture. We need only cell culture. So the virus can be grown only in living media. That means cell culture. So they cannot be grown in normal chemical media. They cannot be grown under normal chemical media. So they can be cultivated only in living media. They are active only in intracellular phase and inactive in extracellular phase. That means when a virus can be active when it is present inside a host cell. It can be active only when it is present inside a host cell. When it is present in outside of the host cell, it cannot be active. So they are active only in intracellular phase. That means they are active when it is present inside a cell. When it is not present inside the cell, they will be inactive. So they are inactive in extracellular phase. So they can multiply, they can feed, they can they can synthesize its own components. All these things happen when the virus is present inside a cell. When it is outside the cell, it cannot multiply, so it cannot grow, it can't do anything, so they are inactive. In extracellular phase, they are called as virion, and in intracellular phase, they are termed as virus. Actually, there is a misconception that we generally term the virus as virus, but it should not be there. When the virus can be denoted in the sense, when the virus is present inside the cell, then only it should be termed as virus. When the viruses are present, when the viruses are not present inside the cell, when it is present outside, that is in extracellular phase, when the viruses are present in outside, they are mentioned as virion, not virus. Once the virion enters into a host cell, then only it should be termed as virus. Clear? When the viruses are in extracellular phase, they are termed as virion. But when the virion enters into a suitable host, then only it can be called as viruses. Right? So coming to the structure of virus. Coming to the structure of virus, the virus contains a nucleic acid covered by a protein coat. You can see in the picture, the virus contains a nucleic acid. This is the nucleic acid. And this nucleic acid is covered by a protein coat. The virus is covered by a protein coat. And this protein coat is called capsid. Since it covers the nucleic acid, since it covers the nucleic acid, it is called as nucleocapsid. Right? So the virus contains a nucleic acid that is covered by a protein coat. That protein coat is called capsid. So this capsid is made up of protein. So that's why it is mentioned as protein coat. And as it covers the nucleic acid, it is mentioned as nucleocapsid. So apart from these nucleocapsids, some viruses may contain the envelope. So this is the envelope. Some viruses may contain envelope. Its envelope surrounds the nucleocapsid. So this envelope surrounds the nucleocapsid. So the envelope is the extra outer covering. It surrounds the nucleocapsid. Actually, the envelope is a lipid layer. It is made up of lipid and two-layer lipids, so that it is a bilayer lipid layer. So the envelope is made up of lipid and it is a bilipid layer. Two layers are there. And in some viruses, the envelope contains some spikes. So these are the spikes. It is the structure which projects outside. So these spikes are made up of glycoproteins. These spikes are made up of glycoproteins. So these spikes are made up of glycoprotein. And these spikes are mentioned as peplomas. These spikes are mentioned as peplomas. Okay, these viruses are mentioned as peplomas. So listen, once again I will tell you, the virus contains a nucleic acid. The nucleic acid may be either DNA or RNA, but not DNA and RNA are not present in a combined form. So both DNA are not, RNA are not present inside the cell. Okay, so it is the nucleic acid, and the nucleic acid is surrounded by a protein coat called capsid. The capsid is made up of protein. Since it covers the nucleic acid, it is termed as nucleic capsid. Apart from nucleic acid, some viruses contains envelope. The envelope is made up of lipids, and the lipid will be two-layered. So it is a lipid bilayered lipid, right? Two layers are there. From the envelope, there are certain structure projects outside. That structure is called the spikes. The spikes are made up of glycoprotein. So all these spikes are made up of glycoprotein, and the glycoprotein is also termed as peplomas. Okay. So coming to the life cycle of viruses. Next one is the life cycle of viruses. So the life cycle of viruses is divided into the following stages: attachment, penetration, uncoating, biosynthesis, assembly, and release. Attachment, penetration, uncoating, biosynthesis, assembly, and release. So first one, attachment. Attachment is the specific binding between viral surface protein and the receptor on host cell surface. Attachment is the first stage when a virus cell when a virus attachment is a specific binding between the viral surface protein and the receptor on the host cell surface. See, when a virus is going to infect a host cell, before that there should be an attachment. So the virus slowly moves to the host cell, and then it gets attached with the host cell receptor. So you know that the cell has a lot of receptor, so it attached with the host cell receptor. So there will be a binding between the surface protein, that is the glycoprotein, the spikes. With the help of spikes or other glycoprotein, it gets attached to the host cell. Then following the attachment, slowly the virus will injects its nucleocapsid, right? Following the attachment, once the virus gets attached with the host cell, then it slowly injects its nucleocapsid. So this may be sometimes happen due to endocytosis or some other mechanisms also. This may be happens due to endocytosis. That means sometimes the host cell engulfs the virus. It automatically goes to the viral and it will engulf. But even though it engulfs the virus cell, there will be attachment happens, and then the virus will penetrates into the cell and it will inject the nucleocapsid. Once the virus enters along with the nucleocapsid enters into the cell, host cell. Then the capsid is removed. The nucleocapsid is removed by the host enzymes or viral enzymes. The capsid is broken, it is degraded, the capsid is removed, and the nucleic acid is released inside the host cell cytoplasm. Then after the release, the nucleic acid is released freely inside the cytoplasm, and the nucleic acid will start synthesizing of its viral particle. It will start synthesis of virus particle. Then after the synthesis of all the required viral particle, then it assembles the cell wall, nucleic acid, protein, all these things, whatever it assembles, synthesized, it assembles all the particle. It is an assembly of synthesized particle, viral genome, capsid, nucleic acid, envelope, everything will be synthesized, and all these things will be arranged inside the host. After all the arrangements has happened, all the parts are assembled, then it will become a full complete virus. Then it escapes from the host cell by just lysing the cell wall. Just lyse, it tears the host cell and lysing, and it will release from the cell with a lot of viruses. A single cell is released to the host cell, and when it is released from the host cell, it will become thousands and thousands of cells will be released, and all the cells will target for the next cell for its infection. So the infection starts continuous. So this is the structure. You can see the virus is getting attached into the cell, and in the next cell it will inject the nucleic acid. The nucleic acid is released. It assembles all the viral particles. Then it assembles, synthesis, the assembles here. Then the virus will be released. The virus is released by lysing the cell. So that's all about the introduction of viruses. If you have any doubt in this, you can ask me. You can text me.